before I begin, I'd like to thank Diabetes SA for giving me the opportunity to present some of the work that we've been doing over at the SAHMRI. So for those of you who don't know where SAHMRI is, we're located right in the middle of the Adelaide Biomedical City. So we're right next door to the Royal Adelaide Hospital, and we're also, our neighbours are also Adelaide Uni, who have the Health and Medical Sciences building, as well as Uni SA, who have their Health and Translation building there. Coming up in the next couple of years, you'll find is SAHMRI 2, where we will house our proton therapy, and that will be our Australian Bragg Centre. Now, SAHMRI houses over 700 medical researchers, spanning from bench to bedside research, and we cover a whole range of different diseases. And just to give you an idea of the research focuses that we have, sorry, I realize there's two screens. These have been divided into several themes. So the first one being SAHMRI Women and Kids, so primarily focus on mothers and babies' health. We also have a strong research focus on Aboriginal health equity. This is led by Professor Alex Brown. We also have a theme called Precision Medicine. This is primarily looking at precision medicine to target diseases such as cancer, the microbiome, and also look at gene editing targets as well. I'm part of the, if I can find the pointer, lifelong health theme. So this is looking at diseases. We focus on improving health for your entire life. So this is divided into heart and vascular health, nutrition, diabetes and metabolism, neurobiology, and mental health and well-being. And our research focuses begins from discovery platforms towards preclinical and hopefully to clinical and translational platforms. Now, if you're interested in hearing a little bit more of our research, we run fortnightly public tours, and you get to see the insides of this building, which looks pretty impressive, I think. All right, so now that my spiel for SAHMRI is done, let's get down to a bit of science. So a brief overview of what I'll be talking about today is I'll touch briefly on cardiovascular disease and give an overview of what it involves. I'll describe why cardiovascular disease is particularly prevalent in the context of diabetes mellitus, and then I'll present some of the data that our group has been focusing on in terms of identifying new therapies that can help treat diabetic vascular complications. The first one we'll talk about is the beneficial properties of high-density lipoproteins, or HDL, and then I'll touch briefly on what I term the ability to micromanage diabetes-associated cardiovascular disease, looking at potential therapeutic targets and diagnostic biomarkers.
I've been part of investigative teams that have put away some of Australia's most infamous serial killers, but I had no idea that Australia's most prolific killer was stalking me. I was lucky to survive the attack, but so many others haven't. This perpetrator remains on the loose, tearing apart families and communities and taking lives indiscriminately. There's no pattern to it. Men, women, young, old, it doesn't matter. We have to do more to stop this reign of terror.
So this was a series of videos that were promoted by the Heart Foundation earlier in the year to highlight the impact of heart disease, to raise awareness, because a lot of people think that heart disease has been fixed, but actually the impact of heart disease still remains in Australia and globally. So 1 in 6 Australians have cardiovascular disease. This totals 4.2 million Australians. And of this, most people over the age of 65 years or over are currently living with cardiovascular disease. This results in the main cause of more than 575,000 hospitalizations in 2016 and 2017, and unfortunately remains still the leading cause of death in Australia, killing 1 Australian every 12 minutes. And in 2017, heart disease claimed the lives of nearly over 43,000 Australians. Now, what is cardiovascular disease? So it not only encompasses the heart, but also covers the blood vessels that bring blood to the different tissues. And depending on where the impact of a blood vessel occlusion happens, this could lead to stroke. Obviously, in the heart, it affects coronary artery disease, and also it affects the lower limbs and feet, and this could lead to peripheral artery disease. Now, the main thing that drives cardiovascular disease is atherosclerosis, and this is the buildup of fatty deposits in the arteries that can lead to the narrowing of blood vessels. And this is a disease that develops over decades, and primarily what will happen is upon injury, the endothelial cell, which is the first layer in your blood vessel, it becomes damaged. This promotes the recruitment of immune cells, such as monocytes and macrophages, that enter the blood vessel, and they start to try and fix the injury. However, they start to pick up lipids, because our diets at the moment are fairly high in fats, and then these develop into fatty streaks, and this could develop, and if it progresses, this could lead to a plaque formation. Now, the bigger the plaque is, the more that your vessel is occluded, and this is what causes the range of series of cardiovascular disease symptoms that we talk about. Now, the initial stages of atherosclerosis is often asymptomatic, and you don't really detect cardiovascular disease until the later stage, and often that is too late. And the initial processes that drive atherosclerosis include inflammation and oxidation. So these are cell injury damages to the blood vessel. Now, there are known non-modifiable as well as modifiable risk factors for cardiovascular disease. Obviously listed here are non-modifiable. So as you begin to age, you have a higher chance of developing cardiovascular disease. Gender is also an important factor, with males being more likely to develop cardiovascular disease compared to females. However, studies have shown that postmenopausal females do then have a higher risk of developing cardiovascular disease. Obviously, things like genetic factors and ethnicity also impact on the prevalence of cardiovascular disease. Additionally, there are modifiable risk factors, and this includes smoking, high blood pressure, physical inactivity, obesity, high blood cholesterol, and also diabetes mellitus. Now, I don't have to tell the audience the global impact of diabetes, and we know that the International Diabetes Federation are projecting 1 in 10 people, or 642 million individuals, will be living with diabetes by the year 2040. Current statistics include that we know that 1 in 11 adults have diabetes, and staggeringly, 1 in 2 adults with diabetes is currently undiagnosed. This has a significant impact on global health expenditure, amounting to 12%, or $727 billion. To date, lives lost globally from diabetes number 1 every 7 seconds, or nearly 5 million annually. And if diabetes was a country, it would be the world's 3rd largest. So this gives you an insight into the impact that diabetes has globally. Now, a little closer to home, 280 Australians develop diabetes every single day. That's 1 person every 5 minutes. So by the end of my talk, another 8 people will have diabetes. Current numbers that have diabetes is 1.7 million Australians, and that's the same as the population of South Australia. And of this, 1.2 million are known and registered to have diabetes, but a staggering 500,000 individuals have diabetes and do not know it yet. And this has a significant impact on the cost of our health expenditure, costing an annual of $14.6 billion every year. Now, if you look at the risk factors for type 2 diabetes mellitus, a lot of them overlap with the risk factors for cardiovascular disease. So these are highlighted either in the blue for non-modifiable, as well as in the red for the modifiable. So taken together, this has a double impact on the prevalence of cardiovascular disease, and in people with type 2 diabetes, cardiovascular disease occurs nearly 15 years earlier compared to individuals without diabetes. People with diabetes have 2 to 4 times more chance of developing cardiovascular disease and stroke, and cardiovascular disease is the main cause of death of up to 80% of diabetics. Furthermore, women with diabetes have a 3 times rate of cardiovascular disease and stroke compared to diabetic men. And diabetes accounts for more than 60% of all non-traumatic lower limb amputations. So not only does this have a significant impact on death, unfortunately, but also patients' quality of life. As I've said, cardiovascular disease is the leading cause of death in people with diabetes, and this is true irrespective of coming from low-income, medium-income, or high-income countries. Some of these deaths, primarily, the majority of this is attributed to ischemic heart disease, so where the tissue becomes, due to the blockage of blood vessels, the tissue does not receive enough oxygen into that particular, into the heart, so this can lead to myocardial infarction. It also accounts for 15% of other forms and 10% of stroke. Now, type 1 diabetics are not exempt from the prevalence of cardiovascular disease. They experience cardiovascular disease a lot earlier, as you can see based on the age range here. 16% will develop cardiovascular disease, 2% stroke, and 1% heart attack. However, these numbers are increased when you have type 2 diabetes. Now, why does diabetes have such a great impact on the prevalence of cardiovascular disease? And this is my one and only schematic diagram for this talk. So basically, as I mentioned earlier, atherosclerosis and cardiovascular disease is a disease that develops over several decades. And there's a wide range of different cellular processes as well as different cell types that help drive this progression. Now, diabetes creates a cellular environment that is pro-inflammatory, with the high levels of high glucose that can trigger the progression of this to go a lot quicker. And overall has a lot of impact in terms of driving cardiovascular disease risk. So some of the known mechanisms that increase cardiovascular disease include disproportionate levels of lipidemia. So these are your cholesterol levels, whether it's good cholesterol, it's lower levels of your good cholesterol and high levels of your bad cholesterol. High concentrations of glucose have a significant impact in the cellular environment of the vessel wall, again similar with high levels of insulin. Endothelial dysfunction, as I mentioned, being the initial layer of the blood vessel, gets damaged very early on, and this has a significant impact on cardiovascular disease risk. Also, cellular processes such as oxidative stress, inflammation, I can never pronounce this word, hypercoagulability, so the ability of clot formation, as well as impaired angiogenesis, which is blood vessel formation. Now, while there has been significant advances in the treatment and a lot of individuals have responded to therapies, people with diabetes still have higher rates of cardiovascular disease compared to people without diabetes. And despite recent advances, many patients do not respond to current treatment approaches, highlighting the need for alternate strategies. So, because atherosclerosis is a disease that develops over decades, we are looking at new approaches to prevent the development of atherosclerosis, identify new ways to detect the disease, as well as novel therapies that can help treat or reverse this process. And this is where our group comes in. So, we are led by Dr. Christina Bursell, and this is a group of postdocs, students, as well as research assistants who are looking at different approaches to address new ways to treat cardiovascular disease and diabetes. We use a multifaceted approach, looking firstly at improved blood vessel imaging capabilities. So if this gives us an insight on what the blood vessels look like, we can then target or tailor appropriate treatments in the future. Identifying new ways to detect the progression of disease early on is critical, particularly in the early stages of atherosclerosis, given that those stages are often asymptomatic. We're looking at exploring levels of ways to increase levels of the good cholesterol, HDL, improving stent biocompatibility. So stents are what happens when cardiologists, if you have a blocked vessels, cardiologists will deploy a stent to open up the blood vessel a little bit more, and there are complications that are associated with that. So we are looking at ways to improve stent biocompatibility. We are also exploring inhibitors of inflammation, given that's the early stage of disease progression. And for the rest of my talk, I'm going to talk to you about how we are looking at ways that we can promote new blood vessel formation. So why blood vessel formation? So that is a critical physiological process. And when the vessel becomes occluded, as you can see here, the tissue becomes ischemic. So it has lack of oxygen, so there is no way for blood to pass to deliver oxygen. So the normal body's physiological process is to try and grow new blood vessels to bypass this blockage and promote and return blood flow. So angiogenesis is the formation of new blood vessels from pre-existing vessels, and it is important, as I said, to bypass blocked arteries. However, in diabetes, this severely impacts the body's ability to promote blood vessel growth. So this contributes to many of the cardiovascular complications of diabetes. And here you can see just a little cartoon of how the collaterals are significantly reduced in a person with diabetes. So therefore, agents that can promote new blood vessel growth in diabetes would be therapeutically beneficial. So this was work that began at the Heart Research Institute in Sydney, and this is just a picture of the group that was involved. Obviously, you can see Chris is here, and when she came over to Adelaide, she offered me a position to come over here to continue some of the exciting work that we're doing. So what are high-density lipoproteins? So I think most of the audience would know that there's good cholesterol as well as bad cholesterol. And the easiest way to talk about this, well, for me to talk about it, because I'm still getting used to it, is that lipid deposition is an initiating process in blood in atherosclerosis. So the LDL, or the bad cholesterol, deposits lipids, excess cholesterol, into the artery walls. But what the good cholesterol does is it takes the lipids from the artery walls and delivers them to the liver to be processed. So there's this fine balance of making sure you've got enough good cholesterol versus bad cholesterol. And that's one of the key things that gets measured when you look at lipid levels. So extensive research has explored the role of HDL because of this seminal study conducted in 1977, the Framingham study, which showed that increasing levels of high-density lipoproteins are associated with a reduced risk of cardiovascular events. So this led to a whole suite of different studies to explore why HDL is cardioprotective. Now, HDL is an inner core of cholesterol surrounded by an outer layer of phospholipids, free cholesterol, and apolipoproteins. An extensive basic bench and preclinical studies have shown that HDL can promote or rescue some of the key processes that are critical to atherosclerosis. This includes its ability to remove lipids from vessel walls. It's anti-inflammatory. It has antioxidant properties. So these are the two processes that are the initiating phases of atherosclerosis. It is also able to prevent blood clots. We have shown that it can also control new blood vessel repair. Now, recent studies have shown, however, that in disease states such as cardiovascular disease and diabetes, HDL loses its ability to promote these functional protective effects. So as disease happens, it's not necessarily so much how to raise the HDL levels, but the quality of the HDL that's flowing in your blood. Furthermore, in the context of diabetes, low levels of HDL is associated with the increased risk of developing diabetes. And a recent study conducted at the Baker Institute have shown that HDL infusions into patients with diabetes can reduce blood glucose level. Furthermore, our group has shown that HDL can rescue diabetes-impaired angiogenesis. So we did this from the bench. We first conducted cell studies where we treated cells with either the good cholesterol or just a control. And what we did was we seeded these cells onto a gel matrix. So this allows the cells to form 3D structures similar to a honeycomb-like, as you can see in this cartoon here. And this is reflective of a cell culture way of promoting blood vessel. And what we did was we found that under normal glucose, cells that were treated with HDL was able to promote these honeycomb networks. And in cells that were treated in high glucose, there was a significant impairment in these cells to promote tubes. However, in the cells that had HDL, these cells were able to start forming these vascular networks again. We then explored this in our preclinical surgical models, and what we did was we induced blood vessel return. And again, you see in our non-diabetic animals, infusions of HDL can promote blood flow return, as seen in the red limbs. In our diabetic animals, you can see that this blood flow return is significantly impaired, as you can see by the blue and green color, as opposed to the red increased high blood flow. And excitingly, we found that in animals that receive HDL, there was a significant increase in the red color you see in our foot. So this shows for the first time, not only in our cell culture studies, that we can also see this in our preclinical models. We also conducted a second study using our wound healing model. And the reason we do this is we know that diabetics have severely impaired wound healing, and this is associated in part due to poor blood vessel formation. So what we found was that you can see here, in our non-diabetic animals, HDL was able to promote wound closure, and diabetes severely impairs wound closure, as well as blood flow return, as you can see by the blue and green when we measured blood flow reperfusion. However, in the animals that had the daily topical application of HDL directly onto the wound, this promoted not only both wound closure, but also increased blood flow return, as you can see by the green and red here. So these findings suggest that HDL has strong therapeutic potential for the treatment of diabetic wound healing and vascular complications. And excitingly, we are currently conducting the first human proof of concept trial that will test topical HDL treatment on wound healing in patients with diabetic foot ulcers. This is driven by Dr. Christina Bursell in collaboration with Professor Rob Fittrich, who's at the Royal Adelaide Hospital, as well as Steve Nichols, who's an international a cardiologist and an international leader on HDL. And this is supported by Diabetes SA. So switching gears slightly, or I should say, watch this space for the outcomes of that. Switching gears slightly, I'm going to spend the rest of my talk talking about micromanaging diabetes-associated cardiovascular disease. And I've termed this micro because we are interested in microRNAs. And this is driven by two very, talented students looking at the role of microRNAs as a potential therapeutic target. This is Emma Solly, who drives this proportion of the project. She is a very talented research assistant that I hired and has now decided to do a PhD, good or bad, depending on how she goes, with me. We're also looking at microRNAs as a potential diagnostic biomarker, and this was our honors student, Katie Morrison. So I'll present some of the work that these very talented ladies have generated. Now, before I do that, I thought I'd talk about microRNAs, and if Matthew could help me out at the back there.
It sounds like a recent discovery in biology is proving to be one of the most important scientific breakthroughs in the last decade. Even though they are only tiny molecules, leading scientists are calling microRNAs the master maestros of the genome. MicroRNAs are quite abundant in cells and have a major influence in cell biology. A single microRNA has the ability to orchestrate the expression of broad networks of genes instrumental in normal cell function. As a microRNA regulates the function of several messenger RNAs, it finely tunes a biological pathway, which can profoundly affect cellular activities. As we discover how microRNAs are involved in biological pathways, we'll also be discovering how their dysregulation leads to disease. Typically, when a microRNA is dysregulated, the pathways that are the targets of the specific microRNA go awry, which disrupts the delicate balance and harmony of normal cell function. MicroRNA medicines act by targeting dysregulated microRNAs, resulting in the normalization of genetic pathways and thus halting the disease process. The prospect of targeting microRNAs is very promising because it opens the possibility to a new class of therapeutics and a unique approach to treating diseases. The field of microRNA discovery and therapeutic development is at a tipping point and is growing exponentially. We at Regulus have developed innovative chemistries and acquired a deep understanding of the biology behind diseases caused by microRNA dysregulation. Regulus is uniquely positioned to lead the development of a new class of high-impact medicines based on microRNA. Regulus, targeting the pathways of human disease. To learn more, visit our homepage at www.regulusrx.com.
So I'm not sponsored by Regulus at all, but I thought that was a really cool video to explain what microRNAs are, rather than my fairly dry explanation from a science point of view. So we were interested in what microRNAs regulate diabetes-impaired blood vessel formation. So in addition to what we showed in terms of HDL promoting blood vessel when it's needed, we also showed that HDL can inhibit blood vessel formation when you don't want it in the context of cancer. So we did an initial screening to identify any novel microRNA targets, and what we found was that there were two microRNAs that were regulated by HDL under both angiogenic stimuli. Now, this gave us a bit of hope because MIR27B already has known pro-angiogenic properties. However, very little was known about MIR181C. So the first thing we did was we went back to our cell culture assay, and what we did was we treated these cells with little chemicals that can either inhibit miR-181C. So what you found was that when we inhibited miR-181C, there was an increase in the ability of these cells to promote tubes. However, when we overexpress miR-181C, so we increase the levels of miR-181C, these cells then lost its ability to form tube formation. So excitingly, we showed for the first time that miR-181C plays a role in blood vessel formation. We then wanted to see what happens in diabetes. So we looked at miR-181C levels in cells that were treated with high glucose, and we saw that miR-181C levels were increased, and we also saw this in our diabetic mice. So suggesting that miR-181C levels are elevated in a diabetic environment and may play a role in the impact of diabetes in terms of reducing blood vessel formation. So we wanted to see what happens whether we can rescue diabetes-impaired blood vessel formation by inhibiting miR-181C. So this is work that Emma has done. Again, we see that our diabetic controls have low blood flow return, as you can see by the blue and green in the limbs. In our negative control animals, we also see that this has a significant impact in diabetes. However, when Emma injected a target to inhibit miR-181C, we can see that blood vessel return in these legs are significantly higher by the red and green. So we've proposed that miR-181C may be a novel target for therapeutic stimulation of angiogenesis in diabetes. And now I know I got the 5-minute mark there, so I'll just try and whip through the next little bit. So in addition to finding new ways to treat, we wanted to explore new ways to prevent and detect atherosclerosis and cardiovascular disease. So if we can detect it sooner, the sooner we can treat to try and reverse this impact of cardiovascular disease. And we were particularly interested in the impact of cardiovascular disease and diabetes in Indigenous Australians, because cardiovascular disease is the greatest promoter to premature death in the Indigenous population, with the 35 to 44 age group being 9 to 12 times more likely to die from cardiovascular disease. And this does not change as you age. And they're 3 times more likely to suffer from major coronary events. Now, why are Aboriginal Australians so compromised? They have 3 major risk factors that drive cardiovascular disease. They're more susceptible to diabetes, they have higher burden of inflammation, as well as lower levels of HDL. Now, if you look at the impact of diabetes, it's pretty much a mirror image of the cardiovascular burden. And again, Aboriginal and Torres Strait Islander peoples are 3 times more likely to develop diabetes and experience nearly 6-fold higher hospitalization rates due to diabetic complications. Now, in an effort to try and close the gap, we wanted to identify whether there were genetic and/or biological predispositions to cardiovascular disease and type 2 diabetes, and also identify new and improved ways to better detect disease severity. Now, jumped ahead. Now, in addition to it being a therapeutic target, microRNAs are very stable in the circulation. This gives them a potential avenue as biomarkers for disease, and it can be transported either on its own, in a protein complex, or microvesicles or exosomes. And circulating microRNAs have been shown to correlate with disease, and importantly, HDL are known carriers of microRNAs, and and HDL-associated microRNAs have also been shown to be different in different disease states. So this was a collaboration with the Aboriginal Health Theme, where we had access to patient samples from the Aboriginal Diabetes Study. This is headed by Professor Alex Brown. We had, for our patient cohorts, we had non-Indigenous without diabetes, Aboriginal without diabetes, with diabetes, and with diabetic vascular complications, specifically PAD. For these, we had plasma samples and isolated the HDL fraction, where we measured miR-181C levels. We also then took the HDL that we isolated and added them to cells because, as I mentioned earlier, disease states such as cardiovascular disease and diabetes tend to impair the function of HDL. So we wanted to see whether the HDL from these individuals had an impaired capacity to form tubes. And this was work that was done by Katie Morrison. So what Katie found was that, interestingly, overall, the Aboriginal population had lower miR-181C levels in the plasma. However, none of these were significant. And bear in mind, this was a small pilot study of 7 to 10, so we obviously need more N numbers. Interestingly, however, we found that in the Aboriginal population with diabetic complications, there was a striking elevation or increase in HDL-bound miR-181C levels. So we were quite interested in that because miR-181C has the ability to inhibit blood vessel formation. So whether HDL is carrying this and delivering it to sites and allowing the MERS to kind of have a target in the tissue would be quite interesting. So we did this in a cell culture study, again going back to our tubes in culture, and this is our HDL as a positive control based on our previous studies. What we found was that in our non-Indigenous and in Aboriginal participants without diabetes, these cells were still able to form tubes. Interestingly, we also saw that in the HDL from Aboriginal with diabetes, they were still able to form these little vascular networks, as you can see here. However, in the HDL from patients with the diabetic complications, these cells had an impaired ability to form tubule networks. So this suggests that potentially HDL is carrying this anti-MER, this microRNA, and having an impact functionally in the tissue. But obviously, again, this is still very early work, but we're excited with some of the findings from this. And we postulate that elevated HDL miR-181C may be a novel biomarker associated with diabetic complications. So this is just the tip of the iceberg. There's a lot of questions based on these studies. We want to extend and see if miR-181C can affect other things beyond blood vessel formation. Can it rescue other diabetic complications such as wound healing or myocardial infarction? Is miR-181C a biomarker in the non-Indigenous population? Are there any other microRNAs that we can use as potential targets? So some of the take-home messages are that we know diabetes increases cardiovascular risk. Many do not respond to current treatments, highlighting the need for novel approaches. I've shown you today that HDL rescues diabetes-impaired angiogenesis, and we currently have a clinical trial to look at HDL in diabetic foot ulcers. And also, we postulate that microRNAs can provide a two-pronged theranostic approach, both as a diagnostic biomarker as well as a therapeutic target. So we'd like to say that our research is aiming for a bench-to-bedside approach. So I guess our HDL levels, our HDL studies are currently at the development stage of clinical trials, and our miR-181C is kind of a little bit further back, but hopefully with the potential to progress in the future. And finally, I'd like to acknowledge everyone involved in these studies. I am just a representative of a bigger cohort, as you can see, and these are the main people involved, and also all the funding bodies that have contributed to getting this research across the line. Thank you.
With the use of the HDLs for the diabetic foot ulcers trial, is it relatively easy to produce HDLs? Is this going to be a cost-effective treatment? And do you foresee any side effects? Because that's pretty amazing differences for a relatively easy and, what I imagine, low-risk treatment to the patient as opposed to a systemic treatment.
Yep. So for the clinical trials, we are collaborating with CSL, who can generate HDL. We generate it in the lab as well, but they were at the point where we can treat it to patients. But there are companies that develop HDL as well as mimetics that mimic some of the key proteins that could affect that. So in the future, I think it is, I would say, fairly easy, given that HDL is already present in the body. It's not likely to drive any immune responses as well. So that's probably the angle that we're going with.
And the HDL that you would treat with is generic. It's
generic, yeah. So it's generic, and it's not from the patients, because I think we are finding that current HDL raising therapies have not yielded the same benefits as we expect in our preclinical studies, and we think that it's not so much the amount you're raising, but the quality of the HDL. And if you're trying to raise HDL levels in people with cardiovascular disease or diabetes, maybe you're raising the wrong type of HDL. They're actually more detrimental to the development of disease. So these are generic from companies that will produce them.